Good afternoon, everybody. My name is Pablo Abelchauke, and I'm a training and outreach coordinator for HTA Bionet, based at the Central Node, which is based at the University of Cape Town, South Africa. And I'm part of the HTA Bionet Seminar Coordinators Task Force, which organizes monthly webinars for HTA Bionet and the African community. But obviously, we broadcast to every part of the world. And for the July webinar, we are pleased to have Associate Professor Matt Ritchie, who is in Australia, so there's a time difference as well. It's 11 p.m. that side, and it's 3 p.m. In South Africa, and it's obviously other different times in different parts of the world as well. So we are grateful to have Professor Matt Ritchie, who's going to be speaking about putting the R in bioinformatics with Bioconductor. This talk will be for 40 to 50 minutes, and we'll have 10 to 15 minutes at the end of question and discussion. So if you can hold your questions until the end, that would be really appreciated so that we don't disturb our speaker. So kindly type your burning questions in the chat box, which is on my left. Or if you really have a burning question, there's a human-looking icon on top that you can press that is going to show Professor Matt Ritchie that you have raised your hand. I'm just going to quickly read a bit of the abstract as well as give the CV that Professor Matt has illustriously in his life. The title of the talk today is Putting the R in Bioinformatics with Bioconductor. Bioinformatics analysis and software underpins each and every new discovery made using genomics data and medical research. This talk will give an overview of recent advances in gene expression technology and showcase their work in developing new statistical methods and open source software for use in expression analysis available through the Bioconductor project. Improved methods for prioritizing genes and pathways, as well as new plotting capabilities that allow more intuitive exploration of both raw and summarized data, will be presented in today's webinar. Just a quick summary, because I mean, as most of our speakers have long CVs and have done amazing work. You can find some information on their website from where he particularly. I'll just read a few lines about what Professor Matt Ritchie has done. He's an associate professor at the laboratory, or rather at the Epigenetics and Development Division of Walter and Eliza Hall Institute of Medical Research in Melbourne, Australia. Professor Matt Ritchie is an experienced computational researcher with skills in analyzing high-throughput sequencing data and developing open-source software for bioinformatics analysis. Through close collaboration with bench scientists, his analysis have revealed new insights into epigenetic processes, apoptosis that have been published in leading journals, including Cell, Cancer Cell, and Genome Biology. He has been an avid R user since the year 2001, so he has more than 20 years in the field. Without further ado, I would like to give the mic to Professor Matt Ritchie and say, go ahead and give your presentation.
Thanks, Pablo, for that kind introduction and for the opportunity to present to you all. So yeah, my name's Matt. I'm very excited to be here with you today to tell you a bit about using R and Bioconductor for genomics analysis. So, how do I advance? Here we go. So to give you an outline of the talk, the first part's just a very brief history of modern genomics technology that's used for profiling gene expression, which is something that we do a lot in my lab. The second component is about what on earth is R got to do with this, and how has Bioconductor come into this situation. And then finally, I wanted to introduce some of our software for performing gene expression analysis. So if we go back not so far, about 25 years, people started getting exciting about measuring gene expression in a genome-wide fashion. Before that, you'd have to do things a gene at a time, and it was very laborious. But in December 1996, two papers came out, one in Nature Genetics and one in Nature Biotechnology. One was from Joe DeRisi, and that was kind of advocating for a homebrew printed microarray platform that you could make yourself in your own lab. And the second was a commercial option from a company called Affymetrix. And these two papers really, got people pretty excited about the sorts of things you could do if you could start measuring samples, every gene, expressed in a sample and apply that to your favorite model system or disease system. And some of my favorite examples from those early studies are when people started thinking about, oh, well, when we watch cells go through the cell cycle, it'd be cool to be able to see the waves of gene expression. Genes go up and down depending on where they're implicated in the different phases of the cell cycle. And for disease researchers, it was very exciting to be able to profile disease tissues such as breast cancer tissue, and then notice that there were very distinct gene expression profiles in different subtypes, and that these became kind of molecular hallmarks of different subtypes that then could be used to target treatment to different patients. And a lot of other companies got involved and interested in selling microarrays to the world. So companies like Agilent, Nimblegen, and a company called Illumina, which is also very big in genomics, came along and in 2004 released a beta array product for measuring gene expression. So what all of these platforms have in common, if you put down a little cartoon of a given gene, is they have probes designed to be complementary to the gene sequence that are on the array. They might be short probes, oligo probes in the case of a lot of these commercial platforms, or large cDNA clones that are spotted on the homemade platforms. And basically, you're measuring gene expression by looking at hybridization between the sample that gets some fluorescent label attached to the RNA that then hybridizes to these probes on the arrays. And then, of course, there's probes for all the genes in the genome that you're studying. You might notice, though, that this approach has some shortcomings. You're not covering the whole genome. It's sort of a genome-wide approach, but it doesn't cover all the bits of the gene. So in the mid-2000s, some of the companies, such as Affymetrix, got excited about, well, we could tile the whole genome. We could lay out these probes all over the genome and then really be genome-wide about the measurements we're going to create using this platform. But I'm sure, as you'll appreciate, that the genome is a big place, and so to do that efficiently, you actually need lots and lots of these chips. So, for example, to profile a human genome-wide using this tiling approach, you need 14 chips. So, here we go. Here's one sample. Here's a second sample. Oh, we've got some replicates. So here's only 4 samples, and you need bucket loads of RNA to be able to profile genome-wide using these tiling arrays. So they never really took off, but of course in 2008, second generation sequencing became available, and people started applying this to measuring gene expression. So now we get away from having to define probes that target specific genes, and now we can basically just fragment the RNA in a sample, process it on one of these high throughput sequences, and then try and reconstruct the transcriptome by piecing together lots of these little short reads. So there's my potted history, a short history of gene expression profiling platforms from the mid-90s to present day. So you might be wondering, well, what on earth has R got to do with any of this, and what is Bioconductor? Well, all of these technologies share some features. You have to measure gene expression. So for the microarrays, it's by fluorescence. So you end up with a large matrix of expression values, and you have to process those values carefully to be able to get some insights into which genes are changing over your experiment. So at the beginning, people would just print out the plots of their favorite, the genes that they were profiling. So some of the early studies, and they just send them around the lab, and people would score them as are they interesting or not. And then, the genes that they would highlight in their study would be based on this kind of manual curation of interesting patterns that people were seeing from these plots they were eyeballing. But that approach doesn't scale particularly well. So what has R got to do with any of this? Well, R, of course, is a statistical programming language developed along a very similar timeline to some of these platforms. So R itself began development in a research department at the University of Auckland in 1993, was released as open source in 1995, and then version 1 in February 2001, And then Bioconductor came along with the goal of using some of this R infrastructure that was being developed to help with the analysis of data, and then apply that to analyzing genomics data. And all of this software is open source, so people can freely download it and use it, which is really cool. And I was really excited to see this article published in the New York Times in 2009, something I could send home to my parents to say, oh yeah, this is what I do for a living. I use R and I analyze data. And the gentleman on the left there, Robert Gentleman, is one of the founders of R who started developing this new programming language. And Ross Ahaka is there on the other side. So these two researchers developed R and released it for others to use. And at the time, Robert was working on genomics data and really saw a need to develop an ecosystem for allowing the analysis of these new high throughput data types. And so the Bioconductor grew from just a handful of packages in 2001 when it was originally released to now over 1,900 packages in 2020. And these packages can do many different types of analysis. A key strength is being able to analyze gene expression data, which is some of the technologies that I've highlighted previously. To be able to also analyze ChIP-seq data, microarray data, data from FACS assays, all kinds of assays that people routinely do in the lab that generate lots of data that need to be wrangled and handled carefully to be able to make some good inferences from those data. And so if you go to the Bioconductor website, you can learn a couple of interesting things. The first one is there's actually a conference next week. I think the registration is just sold out. It's a virtual conference, but there's an annual conference where people get together to share their expertise in developing open source software for genomics. You can find out how to install the software, how to learn more about using the software. And there's also special resources for developers to help people develop their own software when they find a niche that hasn't been met by many of the other packages that I mentioned that are available. So Bioconductor has lots of users. It's downloaded heavily and has lots of citations. And I guess that's really testament to the many diverse and useful analyses that you can make using this great ecosystem of tools for analyzing biological data. And there's more than 1,000 developers worldwide who develop new software packages that they then distribute for others to make use of, to share that expertise with others. Some other cool features about Bioconductor that make it a very stable platform for data analysis is that there's a cycle each year of a stable release version of the many software packages that I mentioned, and also a developmental version where new packages go to be tested and new features on existing packages go and they undergo some testing. And then twice a year, the developmental version becomes a release version. And people can start working away with a newer version of R and a newer version of Bioconductor to have all the great new features that have been developed in the intervening 6 months. Another thing that adds stability to the software and a little bit of extra quality, extra seal of approval to it is that all the software is built across multiple different operating systems so that you can know it works on Windows, on Mac, and on Unix. And new packages also undergo New packages also undergo review, so code review to try and make sure that they're well documented, that they adhere to good standards and are likely to be reusable by others. So other features of Bioconductor that make it really useful are the way that people's code is packaged up into distinct little packages, such as the little logos that I showed on the previous slides. So, for example, there are packages that specialize in handling DNA sequences, short read data, packages that are specialized in handling data once it's been aligned to a genome, such as RSAM tools and genomic alignments, packages that help you go from those alignments to then having gene level information so that if you want to do a gene expression analysis, you can go from those alignments to having gene level counts that you can take further and then analyze to find interesting genes And there's, of course, many other packages that specialize in other different things. Another key feature that makes the software ecosystem really useful is that there's encouragement of people to organize their code around key classes and reuse those classes so that packages can understand what they'll be getting for a particular type of analysis. If you're dealing with sequence data or if you're dealing with gene expression data, there's a certain format that takes that you can assume the data will come in, and you can then take that structure and do some new things with it using your code. And so this helps ensure robustness, and it also helps having lots of tools that can assume you're dealing with the same type of data and apply different approaches for looking at that data. So some essentials in Bioconductor that's probably good to know about is the standard representations for DNA sequences in the biostrings packages, for aligned data and genomic ranges packages. And here's some examples, some code to explore some DNA sequence data. And it doesn't give you all the DNA sequence information because that would be quite a lot to print out, just gives little subsets of that. Experimental results are organized in packages such as summarised experiment, which summarize the various information about samples, about the genes, and also about the expression levels that you've got from the experiment that you've run. And if you have data from obviously technologies that produce multi-omics, there's other packages such as multi-assay experiment that can be used. And then further downstream to annotate your data, there's packages that help with that as well, such as various organism packages for dealing with gene level data from different species, amongst other things. So here's an example of trying to get some annotation information out to annotate genes and the code that would run to do that. And here's a nice overview of what a summarized experiment class looks like. So when you have gene expression data, we can think of the assays as the measurement that you get from your experiment, where you've got genes or features in the rows. There's particular annotation information, such as gene IDs, about those rows of gene information that's useful to hold in this object, and also information about the samples. So what are you actually assaying in your experiment? What tissue did it come from? Was the sample from disease tissue or healthy tissue, amongst other things? So this is just some examples of some of the classes that Bioconductor software organizes itself around. And there are many others, for single cell data and other types of data. So that's my very brief overview of the first two parts, modern genomics technology and what Bioconductor is. I'm happy to pause now, though, if anyone has any questions. I can't quite see where the chat bar is, but if there are any questions, if now might be a good time to just pause and catch our breath.
Okay, thanks, Matt. I'm looking at the chat box. There hasn't been anything typed so far, so I think you can continue.
All good. Then I'll continue.
Oh, someone is
All right, it was clear. That's great.
Someone is typing. So, pardon, can you wait for like a few minutes? Nahal, are you typing a question? I see Nahal is typing. Okay, I think let's just give it a few seconds and see how it goes, and then we can continue. Oh, no. So Nahal is saying we are good. So thank you so much. Thank you. You can go.
No worries, just a chance to have a glass of water. Thank you. So I'm going to move on now to some of our research. So some of the tools that we've developed over the past 5 years to try and help with gene expression analysis. So specific research topics, and then also an overview of some software that was developed locally, first for microarrays, and now more recently that also has been adapted to work for RNAseq. So for RNAseq data, just a very brief overview. So what we're doing is we're applying high throughput sequencing technologies such as Illumina, next or second generation sequencing, to study gene expression. There's many applications and many cool questions you can ask of this type of data, but the principal one that my research looks into is the question of differential expression. So so we have our lots of short reads that we align to the genome. We then go through and count up where these reads fall over exons for a given gene, and then we end up with a total count for that gene that we pop in our gene expression count matrix here. So we've got genes as rows, and we've got samples as different columns, and we've got a blue group, and we've got an orange group. And for a differential expression analysis, you're going through this matrix row by row, and you're testing a null hypothesis that there's no change between the blue and the orange groups. And so there are lots of methods out there for doing that, and we have our own methods that I'm going to promote to you today. And so one particular software that is really useful for this is called LIMA, stands for Linear Models for Microarrays originally, and has the past few years also been adapted to work for RNASeq. So the key steps in like a gene expression analysis are, firstly, you've got to get that count matrix in, ready to do some analysis on it. So the key part is a data import step. Subsequently, you want to assess, is your data of good quality? Is it worth taking the analysis further? And that involves a lot of different diagnostic plots and looking at the data in different ways and seeing how the samples behave. And if you're happy with how all that looks, then another follow-on step for a differential expression analysis is to fit a linear model to each gene in your data set and basically estimate the gene expression changes between the different experimental groups in your data set. And finally, you might want to do some gene-seq testing where you do a test not just gene by gene, but now at sort of a pathway level. And so LIMA is developed in Melbourne here in Australia. It's a great local product, and Gordon Smythe has been working on it for many years. And as a PhD student and a postdoc and someone who now runs my own research group, I've also added to LIMA in different ways over its history. So LIMA is able to produce lots of really cool plots. So the plots on the left are some plots for the QC of your data, assessing, well, how the different samples looks, what are the distribution of the expression values, what is the mean variance relationship in your data set, how do different control genes look in your data set, and how do the samples relate to each other. So the plots on the left are super useful for getting a picture of the quality of the data, and the plots on the right are useful for summarizing the results of your analysis. So, what are the top genes for your differential expression analysis highlighted in red? What's the overlap between the different comparisons you've made? Maybe we can make a heat map or do some kind of pathway analysis to look for enrichment. Of course, there are lots of plotting capabilities outside of LIMA as well for plotting your data in R as well. There's heaps of flexibility in how you plot your results, which is a really cool feature of having code available in R and Bioconductor. So at the heart of LIMA are some really neat statistical methods that essentially for every gene in your data set, you're fitting a little linear model that looks like this. So G is for gene, Y is your expression data, X is your design matrix, and beta are the coefficients. Often they're estimated log fold changes between the different experimental samples that you have, the different groups in your experiment, the blue group versus the orange group. And then there's some variance information explained. And the core parts of LIMA, which the LIM is for linear models, is basically fitting these models. Borrowing information refers to the fact that we estimate these linear models over thousands of genes and generally over only a few samples. So the estimation can be improved by looking at how the variability, the distribution of that variability across all the different genes and doing some squeezing to a kind of a common value. Another cool aspect is quantitative weighting. So you can have weights in these regression models and do weighted least squares. And so that allows you to allow different observations to have, allow for the imprecision, that some measurements are more variable than others. And I'll touch on that as some of the research topic that I mentioned later. And that also feeds into being able to model the variances in different ways. So you can fit different models that allow different variance structures on the gene expression values. And of course, the data preprocessing that I mentioned before, which is really useful to just set all this up so that you can go through and reliably fit these linear models. And at the end, you get out some estimated coefficients, and you're testing whether those are 0 or not. So over the remainder of the talk, I'm going to tell you about probably only 2 topics. I think I don't have time for the third one. The first one is methods for dealing with experimental noise, which is basically building on this linear model approach and fitting these linear models in slightly different ways to get sample level estimates of variation that you can feed into the analysis. And the second is some tools in a package called Glimmer for allowing you to plot your gene expression results in a way that's really, I think, useful to the end users of these analyses. It's fun for biologists to be able to explore their data in more detail, and Glimmer allows you to do that. The third topic that I think I don't have time to speak to you about today is EXE, which is software for pathway and gene set testing to allow you to do a great job on testing at the pathway and the gene set level using an ensemble approach, which is basically looking for a consensus across lots of different algorithms for doing gene set enrichment. So this one, last topic, we're going to skip to make sure we don't go over time, but the first 2 I'm going to spend the next little while discussing with you. So the first topic relates to modeling variation, and a common question people have when they're trying to do a differential expression analysis is, all right, what genes are changing between my mutant? So in this case, our collaborators have knocked out a gene called SMOOCH-T1 in some neural stem cells. And they wanted to compare the gene expression between the wild type samples and the mutant samples that are colored here on this multi-dimensional scaling plot in different colors. And the MDS plot, or a PCA plot, is a dimensionality reduced form of your data. So instead of having 10,000 dimensions, because you're looking at 10,000 genes, we distill that down into a lower number of dimensions that explain most of the variation in our data. So here are the first 2 dimensions that explain the most variation. We do see good separation between these 2 different groups, the red mutant group versus the black wild type group, color-coded in this plot. But you can see that one of these samples is really quite different to the others. One of these mutant samples, sample 6, is quite different to the others. It's a bit of an outlier. And this is going to cause headaches. If we do our differential expression analysis, we basically don't find too much because there's not a lot of consistency between the replicates, which is what we're trying to look for, consistent gene expression changes that will be thrown out by this 6th wild-type sample. So how do we handle this situation, which happens quite often, I find? One option is to tell the researcher to go back and do the experiment again, which is quite expensive and often will make them quite sad, particularly given that the sample collection can be very time-consuming, and the samples might not actually be available anymore. They might be a precious sample and they've used them all up. So really what you have is what you have, and you've got to do the best with that data. A second approach could be to try and identify samples that look like outliers and throw them out. That's quite ad hoc, and it relies on you being able to identify those samples, which could be tricky. And the third would be if you had a large sample size, which we don't, we only have 6 samples, you could think of using a robust method. And with this setup, basically, you're going to use the residuals that you get to identify the outliers and then downweight those in the analysis systematically. But typically, we don't have a large sample size in a lot of RNA-seq experiments. So our research problem was to try and solve this. And so we did this by devising a method to deal with sample level variability in an integrated way as part of the analysis. And our hypothesis is that if we downweighted observations from more variable samples, this would really help the analysis. So what we're doing here is trying to weight samples in different ways. So a typical analysis, if we treat all things as being equal, the gene expression measurement from sample 1 and sample 2 and all the other samples will be given equal weight for a weight of 1. So that's the usual way gene expression analyses are done. But we thought it'd be cool if you could actually estimate the variability in our data set. And in this situation, the 6th sample, remember, that was quite different to the others. So you'd say, well, that one's probably more variable. It's actually probably less precise, the measurements from that sample. And ideally, if we could have lower weight to that sample and higher weight to the samples that are more precise, we might do a better job of modeling and getting our estimates of gene expression. So that the deal here is that we want to give more variable expressions lower weight in our differential expression analysis where we fit our linear models. And so we go about this by, instead of just fitting the model row by row, which is what I mentioned LIMA does in the introductory slide, we now also fit a model down the rows at the sample level to get a sample level estimate of variation as well. And so this is the variance modeling that I was mentioning as a key component of LIMA, fitting these models that allow the variability to be different between different observations. And this is something that already goes on in LIMA for analyzing RNA-seq data. And this is a method called VOOM, which deals with modeling the mean variance trend that you see in RNA-seq data when you transform it on the log scale. So this decreasing mean variance trend, which is quite typical, the trend might be more flat or more steep depending on the data set. Each point here is a gene, where you can see there's quite a systematic downward trend. So basically, the genes with lower expression are more variable than the genes with higher expression here. And essentially, this approach uses this lowest line, this smooth line here, to get observational weights, and then it puts those into the regression model. So on top of this analysis, we also wanted to accommodate these weights and also the variant-specific, get sample-specific weights from our fitting of a model to the column data as well as the row data also. So it's time to have a drink break, and we decided to do an experiment to help our research. And the experiment, rather than mix drinks, as we're seeing here, making cocktails out of at a bar, we're actually mixing RNA samples in the lab to be able to simulate data that's actually more variable. And so our approach to this was to take two cell lines and take the RNA from those cell lines and then mix them in various proportions. And so here we've got a red cell line and a blue cell line. They're quite different to each other. And then we make these intermediate mixtures of these cell lines. And so if you're comparing gene expression, looking for like a differential analysis, you can imagine comparing the red sample to the blue sample gives you the most changes. These samples are pure. And then if we compare these intermediate samples, these ones are more similar. So there's more subtle. The gene expression changes should still be there, the same as the blue to the red, but they're just more subtle. So we took these samples, we made 3 replicates of these, each of these cell lines. And then to the second replicate, we kind of heated it up and tried to degrade the RNA, which might seem like a really strange thing to want to do, but we're trying to simulate having a sample that's going to be more variable, different to the others, poorer quality, the sort of sample that if our approach works well, we're going to downweight in our regression analysis. And then, of course, we put it on our sequencer, we got our reads, and now we've been able to get a count matrix to feed into our gene expression analysis. And the first plot we make here is much like before, our MDS plot to look for the relationship between these different samples. The round points are the regular replicates that haven't been degraded, and the triangle is that second replicate sample that was split in 2 that was degraded by heat treatment. You can see, interestingly, that the triangles don't necessarily sit away from the other samples. So we've got the red sample is one pure sample, and the purple sample is the other pure sample, and the green and the blue are the intermediate mixtures. And so they kind of sit where you'd expect them to be, given that they have increasing amount of one sample as we go from left to right in dimension 1 on this MDS plot. But yeah, the triangles, sometimes they kind of sit on the edge of these little clusters, but other times they can be right in the middle. So it's actually quite tricky to see these degraded samples, not as easy as it was to see the outlier sample in the first example that I gave you. So if we fit our model that tries to estimate sample-specific variation, which is again fitting a model down the columns as well as across the rows, you can see for our replicate where we actually degraded the sample, that was replicate 2, it does the right thing. It is systematically downweighting these samples, which we know to be degraded, which is quite reassuring and a good sign that this approach might be useful in practice. And if you go through and apply these, either these VUM observational weights or the combined VUM weights with the sample-specific weights, and compare that analysis with an analysis where you drop the sample replicate 2 that's known to be degraded, even though it's hard to pick by eye, we actually know it's degraded, so we'll assume that it's a good idea to drop that one, and compare that to the results where we only analyze the 3 replicates that are of good quality. You can see that the VUM with the sample-specific weights as well performs best in that it gets the most differentially expressed genes and most overlap with the results from the good analysis. And that's consistent as we go from the comparisons between the pure samples down to the comparisons between the more similar samples. So fewer genes are discovered as we make the comparisons more similar, but again, you get more genes that are the right genes when we apply our VUM with our sample quality weighting strategy. And if we go back to the first data set that I showed you where we were looking at this gene SMUG1, which is known to actually regulate imprinted genes and a set of genes called clustered protic adherence. So our collaborators had done previous research where they know these genes are regulated, and we do gene set testing on these sets. Again, we get better results in our gene set testing, smaller p-values, and more differential expression when we use VUM with our sample quality weights compared to just removing the sample, the outlier sample, which was sample 6. So if this approach sounds like it might be useful, if you're familiar with analyzing RNAseq data, you can try it out at home. So VUM with quality weights is the function in LIMA. It's often useful to be able to downweight low quality samples. We've shown that in some of these demonstrations, and we use this approach quite routinely in our collaborative analyses. And the nice thing about LIMA, as I mentioned earlier, is that it can incorporate these weights at every stage of the analysis. So in the differential expression analysis and also in the gene set testing, which is quite a unique feature. And this delivers more power, so ideally more significant results that you can pass on to your collaborators. So these data are published, this approach is published a few years ago, and then the LIMA package has a publication as well that you can read more about this approach. And of course, download the package too. Feel free to use it, and if you have questions, feedback, let us know. So the final topic that I'm going to touch on just briefly is tools for interactive visualization of gene expression analysis. So there's a bit of you do your LIMA analysis, you'll get a table of results that you might often send to your collaborators as a spreadsheet. And then often you'll make a plot where you highlight the significant genes. Here's a mean difference plot. We've got average gene expression on the X and the log fold change on the Y. And so these red points highlighted here are some significant differentially expressed genes detected by our analysis. But there's a disconnect between the spreadsheet or the table that you send your collaborators and this plot, and often our collaborators are really interested to find out what some of these genes on this plot are, and obviously it's hard to do to match up the information in the spreadsheet with the plot. So we thought it would be really cool to be able to connect these different displays, connect the plot with the table, and provide some interactivity so researchers can really explore these results in more detail. So this led us to develop a package called Glimmer, which stands for Interactive Graphics from LIMA, and to be able to make some of these common plots from the LIMA package more interactive so people can explore them at their leisure and look up any gene or any sample and to find out a little bit more about their data. The Glimmer package also supports other popular RNA-seq workflows, such as if your differential expression results come from HR or DSeq2, you can also make interactive plots using Glimmer for this type of output. And the results are just served up as an HTML file. So it's highly portable. You can email the results to collaborators, and then they can search away and have a great time exploring their data. So the first little demonstration I've got is for an interactive MDS plot. I showed you a few examples of MDS plots before. Here's an example. You run a line of code GLMDS plot, and the plot you get at the other end is an interactive version of an MDS plot where the user can click on the point and find out a bit more about that sample. For example, if there was an outlier sample, they could learn a bit more about that. They can color code the points by different variables that are available. So perhaps there are different strains of mice in this experiment, different RNA preparation methods, and then you can scroll through the different dimensions and look to see if some of these different variables explain some of the variation. And this is really useful for the analyst to work out, well, what's the most useful linear model to fit? Do I have to add in some extra effects for some of these RNA preparation methods or mouse strain or other things? Are these also affecting gene expression or not? So you can explore that interactively, which is quite cool. The second interactive plot, which is really useful, is to really couple the gene level, the summary level data back to the sample specific data and the table of information that you send to researchers when you've done your differential expression analysis. So here's an example of that. What have we got here? How do I start this? Here we go. This is created by the GLMD function in Glimmer. So here you can see we can look up our favorite gene. There's a search bar, and we can look up the gene that we've knocked out in this experiment. In this case, this is the SMOOCHD1 data that I mentioned before. And you can then also see what the raw log expression values are across the different samples in your experiment. You can then look up your next favorite gene. So you're interested in protocoherins because you've heard that they're pretty interesting and potentially regulated by SMOOCHD1. Well, you can find those in your plot and then again see what the raw or normalized expression values are looking like for some of these protocoherins as well. Imprinted genes are also pretty interesting. So here we go. I'm interested in PEG12, internally expressed gene, and I can find where that is in the plot. And again, see how that looks. And you can then scroll through any other gene on the plot too, and you can see it transitions pretty quickly between different genes, and it's a fun way for researchers to explore any gene that you've served up results for. And yeah, the function to be able to make this plot is GLMD plot. So yes, RNA-seq data can be a really interactive experience if you're happy to try out the Glimmer package. So this is another tool that we've published. It's available freely from Bioconductor. And it's really cool to see some journals actually offering researchers to publish their plots as interactive. So for example, Distill allows you to make main figures of your plot interactive. And you can imagine, how cool it would be to be able to scroll through, a paper figure, in the latest journal article you read and make a query on that data for your favorite gene without having to download the data and, search for it from scratch. So hopefully we're in for a way more interactive future in terms of how results and plots are made for our journal articles. So that's exciting. And I wanted to shout out to the IC package to let you know about that. So here's another really cool way of having a way to interactively explore your data. So the input of this, the IC package is a summarized experiment object. And you can see here plots for dimension reduced form of your data where each point is a sample. You can then sort of arrange other types of plots to show other features of the data. For example, how many reads you have per sample, what the reads are looking like in different samples for particular genes, and so on. And So IC is organized around the Shiny platform and allows you to customize, make highly customized interactive plots from your gene expression results, which is really cool. So that's another fun package to look at if you're interested in going interactive for your gene expression results. So that's about as much as I was going to talk to you about today. So I hope you've got something out of the talk. I've given you an overview of gene expression technology and how that's changed over the past 25 years. And I guess if we were to add to this timeline now, we'd be adding single cell gene expression, we'd be adding long read or third generation sequencing platforms that can also be applied to explore RNA expression in different samples, and how, coinciding with this timeline, the development of R and Bioconductor has come along, and going from just a handful of packages in 2001 to over 1,900 packages now. And I've highlighted to you a few of the packages that have been developed where I work through some of the research in labs at the WeHi in Melbourne. The final thing that I wanted to point out, if you're interested in RNA-seq analysis, is at Bioconductor there's a workflow series. So these are packages whose goal it is to make the teaching and learning of different types of genomic analysis easier. So here's an example of a package, RNAseq 123, which is to try and point out to people the main steps that we go through in a typical RNA-seq differential gene expression analysis. So this package, inside of it, has all the code and the data that you need to go through and reproduce a nice RNA-seq analysis to basically get across the main steps in that analysis. So this is a really good teaching resource. So if you have to teach it or you want to learn it, it can be a fun way to do that. So yeah, that's where I'll end. R and Bioconductor is a really great ecosystem for helping you get genomic data science done. And I just want to finish by thanking the researchers who've helped with this project, particular Charity and Alexi, who helped with the mixture analysis that I mentioned to you as part of our developing tools for modeling variability in RNA-seq data. Sean and Charity worked together on the Glimmer package, which is also a really cool tool for being able to serve up results, and Cynthia worked on the variance modeling work as well. Thanks to everyone else in the lab, our collaborators, and of course Gordon, who's the key researcher behind LIMA over many years, and many other people in Gordon's lab who've supported this work. And finally, I want to thank Yagub and Pablo for the opportunity to present to you today. So thanks for your attention, and I'm happy to take questions.
Thank you so much, Matt. Oh, the raiser the hand. I was about to say, type your questions in the chat box or raise your hand. Lizzie, go ahead. Lizzie? Lizzie, if you're trying to speak, we cannot hear you, or at least I can't. Can you perhaps try, because your mic is unmuted, but we cannot hear you. So perhaps try type it in the chat box as well, if we can hear you. While Lizzie is trying to resolve her issue, I was just going to say thank you so much, Matt, because I understand that it's late in Australia, in Melbourne. It's about midnight now, and I really appreciate you taking time out to give this amazing webinar. To all the attendees, thank you so much for attending. We are now left with about 10 minutes maximum for question and answer and discussion. So if you have any comments, any questions, if you need clarity, someone is speaking. Go ahead. Hi.
Okay, please, can you hear me?
Yeah, I can hear you.
Okay, so good afternoon. Thank you for the talk. My question, it may be a little simplistic though, but I'm from the biology background, but I was wondering, generally, when you are developing these packages for use of analyzing genomic data, I wanted to find out what's the thought process like, apart from the programming and the statistics, such that the packages that you bring out can actually really give good predictions about what is going on at the cellular level or the molecular level. Thank you. I don't know if the question is clear.
Yeah, I guess the process for us is often what's something that's a bit frustrating. So for example, for the Glimmer one, it's like we got a lot of, often our collaborators are like, yeah, what's that gene? You know, oh, that gene there looks, you know, that point there looks really interesting. And so it was really motivated by just hearing the same question over and over again and going, oh, okay, I'm sure the technology exists for us to be able to make that interactive. And I forgot to mention, but yeah, we use D3 and we use JavaScript. And so kind of modern sort of web, web applications, technologies allow you to be able to make these plots. So we thought we should give that a go. And so yeah, I guess the motivation is often, what researchers need, the questions that they're asking, and are there existing tools in Bioconductor to do that? And if the answer is yes, then it's like, great, we can use that. We don't have to create our own software. But sometimes we're like, oh, actually, this is a bit of a gap, and it's a gap that we would like to fill because I think it'd be really useful for people to have. And similarly, like in terms of data analysis, like often it's just like, well, we often see, so the example of the VUM with quality weights is like we quite often would see this situation where there's outlier samples and you're like, well, not really sure why that is. And often when you go back to the lab, they can't say, oh, well, we knew that sample was going to be bad because we went on our lunch break while it was being prepared and sat around on the bench for longer. Often, no, people are super careful about their samples and they have no idea as to why one sample would be different from the others because they're all processed in parallel. So it's really just trying to, come up with an analysis method that will try and get around this problem so that they can still get some meaningful results from the data that they have, and so that they can publish their work. I don't know if that answers your question very clearly, but.
Okay, thank you for that. I see Nehal is typing. Nehal is saying, thank you so much for the presentation and information. We have about 5 minutes left. So if you have any pressing questions or comments, please do speak now or forever hold your peace, or you can send an email or tweet Matt Ritchie on Twitter, or like email him and ask for other questions. I see James has typed a question. I can look at it and see. James Chafali says, can you clarify on the variability, technical noise, batch effect, general sample quality that can be mitigated by your weighing approach? Any plans to validate your approach with more samples considering the small margins in some of the comparisons?
So I guess in terms of batch effects, often they kind of manifest themselves in different ways. So I guess what we're trying to model here is sort of differences in variation, like differences in the variance, not differences in the mean. And often with batch effects, I guess what you'll see in your MDS plot is just very clear separation of samples, say in dimension 1 or dimension 2 or dimension 3, that is based on when the samples were run. And so in that situation, provided that the samples are not confounded with batch, it can be very useful to actually put a batch effect in the linear model because that's a clear effect on the means. And modeling the variances there is probably not going to help. I guess the point about technical variation versus biological versus other types of variation. So we're kind of assuming here that it's sort of on top of the regular technical and the regular biological variation that you'd sort of estimate gene by gene. You're going to have there's something additional that's adding even more noise on top of that you want to try and account for. And so that's what this variance modeling approach is trying to do. just saying, well, we can't really see a systematic differences in the means. If there was, and that was based on something, and there was a few samples in the different groups, then you could actually put that in the design matrix of your linear model and account for it that way, and that would probably be the best thing to do. But here it's like situations where actually that you can't do that because there's not a batch kind of variable that's going to explain it. And certainly there are other tools available in Bioconductor, such as RUV, some of the shields shown on this slide, RUV, and other methods as well that deal with batch effects really well also. So you could consider using those if batch variation is a consideration.
Okay, thank you so much.
Thanks for your question.
Thank you so much for that, Matt. And James, I do hope that answers your question because that was a very significant answer as well. We have about 2 minutes left, and I'm aware of time because of time differences in different places, and some of us have other meetings as well immediately after this webinar. So I will give one more chance, or rather one last chance, for someone to raise a question or comment in the chat box or raise your hand. I'll keep quiet for like 2 minutes or so and see if there's anything that comes up, and I'll close the chat for today. Okay. Okay, I'm very cognizant of time, so I'll just say, okay, someone is typing. So it seems as though as I'm about to close, someone wants to ask a question. Michael, go ahead. I'll give you some time to type, and I'll read your question for Matt, and we'll see if it's a question or comment. But just to say, while Michael is typing, I've been recording this webinar. I'm gonna have to download it and then upload it on YouTube, and I'll share the link with everybody by sometime this week, either tomorrow or Friday. So if you missed any part of this webinar, the webinar will be shared. I'll share it using announced lists, and I'll put it on social media as well. But we do have a channel on YouTube where we share our monthly webinars as well, so you can just go through that as well. And I'll also share the slides and email that Professor Matt uses if you want to get in touch with him about bioconductors and our stats as well. Okay, that's if you cannot ask your question now or you missed any aspect of this talk. Michael says, thank you for the info. I'm actually happy that you have an article guiding us on how to go about this analysis. We need such as students. So that's a compliment and comment from Michael there. Thank you so much, Michael, for that kind compliment.
Thanks, Michael. Yeah, just have a look in the workflow section of the Bioconductor website, and you'll find lots of different examples of that. And so again, you can install the package, and installing this package, you have all the code and the data that you need to be able to go through and do a full analysis of different types of data. And there's quite a few for RNAseq too, so you can pick and choose. You don't have to choose the one I showed. There's others as well.
Okay. Prof. Matt, just with that, is there anything that you have to say before I officially close?
No, just thanks for your attention and thanks for the great questions.
Sure. Thank you.
Happy bye.
I'm sure you're gonna get more users now after this webinar. Thank you so much for your time and just the organization and behind the scenes work, and obviously knowing that there's a time difference between you and us in Africa. We really appreciate the fact that it's now midnight in Australia and you actually stayed up to give this interesting webinar. So thank you so much. We'll stay in touch.
No problem. My pleasure.
Thank you. And to all our attendees, see you next month for a different webinar with a different speaker. I'll communicate that in time. Enjoy the rest of your day, everybody. Thank you so much. Goodbye.